and welcome to today's stem cell webinar. I'm Lisa, your guide for the next 30 minutes of science. We're thrilled you're here with your coffee, tea, or maybe your pipettes in hand for what's going to be an insightful session. Today, we're joined by the brilliant Dr. Jessica Hartman. Her background reads like a scientific highlight reel. Degree from UVA, PhD from Duke, postdocs at both Baylor College of Medicine and Duke. She's led research and development across multiple biotech companies and now plays a key role in designing next-generation cell biology workflows at Cell Microsystems. In short, you're in very good hands. Before we launch into the good stuff, a couple of quick reminders. This webinar presentation will run approximately 25 minutes. We'll follow up with a live Q&A session, so send your questions anytime using the Q&A feature. We are recording today's session, so don't worry if you miss a slide or want to revisit something later. We'll send you the link in a couple of days. With that, I'm excited to hand the virtual stage over to the star of the show, Dr. Jessica Hartman. Take it away.
Thank you, Lisa, for that very nice introduction. Usually only my mother calls me brilliant, so I appreciate the glowing review. I want to thank all of you for joining me today in this brief webinar to learn about how we and our customers have been using new technology to simplify, standardize, and streamline induced pluripotent stem cell workflows. So hopefully in the next 25 minutes or so, these are the learning objectives that I hope you're going to take away from the presentation. I'm going to talk about how you can select your induced pluripotent stem cell colonies based on monoclonality, morphology, or gene expression. I'm going to show you how you can minimize manual handling to preserve clonal viability and colony health, how we automate and streamline our iPSC workflows, and ultimately how all of these things combined can improve the efficiency and reproducibility of these complex workflows across a variety of iPSC-derived cell types. So if you are joining me today, you probably are already keenly invested in induced pluripotent stem cell biology, or maybe you are just dipping a toe into it and you're intimidated by the complexity and the fear factor that some people have around these workflows. Now, iPSCs, which are pluripotent stem cells, have been heralded as sort of a boon or a godsend for personalized medicine, with the idea being that we can take cells from a human being and differentiate them into a variety of therapeutic modalities or to study gene edits or even disease-specific relevance for various individuals. And in this very simplified cartoon, obviously it's drawn as a straight line from the iPSCs to the patient. But as you're probably well aware, that is not a straight line in the actual lab or in any kind of program looking to use iPSCs for these workflows. Just to kind of orient you to what we're going to talk about today, if you're not already familiar, induced pluripotent stem cells are reprogrammed or returned to a pluripotent state from somatic cells. The source of this can be basically any differentiated cell in the body, or it can be a differentiated cell type or cell line if you're not starting from patient-derived cells. The act of reprogramming generates these pluripotent stem cells that can then be propagated and expanded, differentiated into every different cell type in the body. They can also be edited for genetic modulation. And ultimately, the goal is to either use these cells as therapies themselves or to enhance drug discovery to ensure proper medicine or personalized medicine for some patients. So as you're probably well aware, none of these workflows are without their challenges, and they each require a high degree of technical effort and a high degree of proficiency in the lab. There are a lot of challenges of working with these fragile pluripotent cells, the first of which is manual manipulation. The vast majority of the workflows require you to manually interact with colonies in a lawn of cells. Those manual manipulations almost always result in viability issues because the survival rate of cells after these kinds of manipulations is not high, as well as heterogeneity in the population. It's very difficult to ensure fidelity of your cells when you are manually picking them out of a 6-well plate. And because of these issues, manual manipulation, heterogeneity of your population, a high degree of cell death, this has limited the availability of these types of cells in scalable or reproducible workflows, whether it's assay development or scaled for therapeutic production. And as I've alluded to, this is largely because when you do these workflows in the 6-well plate or a 10-centimeter dish, you have a heterogeneous iPSC population like you see on the left-hand side of the screen. You then have to interact with those cells using typically a 27 gauge needle or a micropipetter to move those cells out of the dish and into something else for propagation. So I'm not going to go too detailed into the technology until the end of the talk, but it's important to introduce you to how we do this workflow so that you can be oriented for the rest of the presentation. So rather than seeding all of those iPSCs as a heterogeneous population in a dish, we seed them into our proprietary microwell array, like you see here. There's a reservoir inset into a 6mm cassette, and within that reservoir, we fabricate thousands of individual microwells. Those microwells provide individual growth surfaces for our stem cells, allowing us to look at them independently and perform more automated and standardized workflows with iPSCs as compared to the more manual isolation. The cool thing about the technology is that each of those positions can function independently while sharing a contiguous media volume. So we have all the benefits of a shared culture while also enabling spatial segregation and retrieval of our cells. So in our workflow, once we've identified a colony or a cell of interest, we can actually transfer that growth surface to a new collection plate like you see here and propagate those cells for downstream production. So when you see squares or grids throughout the presentation, that's going to be what we're looking at, which is basically our microwell array. So if we look at the traditional reprogramming workflow, which requires this manual manipulation, you can see what that looks like here. If you've ever done this workflow, you probably feel the pain that we've all felt, which is that it's incredibly labor intensive and inconsistent. When you are doing reprogramming, you have put reprogramming factors into a somatic cell of choice, seeded them as a lawn, and you're hoping to identify putative reprogrammed colonies that are pluripotent. As you can see here in a day 15 image in this traditional workflow, there are colonies that have the hallmark morphology of a pluripotent colony, but they are seeded in a lawn of unreprogrammed fibroblastic cells. And so you have to go in there with a needle, dice up the colony, and transfer it to a new plate for propagation. Typically what that results in is what you see on the right-hand side, which is sort of what I call a bare slash colony, where you have either transferred colonies that are dying or differentiated or polyclonal. And overall, this manual workflow typically has less than a 1% success rate and takes even the most skilled operator upwards of an hour to transfer a single 96-well plate of putative polyclonal colonies. So we wanted to see whether or not we could automate this workflow using our technology and improve the success rates and the ability to generate reprogrammed clones. So what we did, as you can see here, is to compare the traditional 6-well plate workflow on the top with our cell raft array, multi-well array workflow on the bottom. As I showed you previously, again, we have that lawn of unreprogrammed fibroblasts surrounded by TRA160 or putative pluripotent colonies in the lawn of cells. In contrast, using our multi-well array, you can see that each of those colonies are individually segregated in each of those positions. And when we apply that TRA160 dye, looking for reprogrammed colonies, you can see an individual reprogrammed colony that is segregated from the remainder of the unreprogrammed cells. As you can see here on the right-hand side of the slide, we actually quantitated this. My poor research associate went into the lab and manually counted every single TRA160 positive spot in the 6-well plate compared to the automated method using our software to calculate TRA160 positive rafts on the air system. And what you can see here quite clearly is that just by seeding the cells on the array from the same exact electroporation, we were able to recover almost 2 times the number of reprogrammed colonies compared to the standard 6-well plate format. In addition, the other benefit of the technology is not just that we can find these individually segregated clones, it's also that we can track and trace the reprogramming efficiency from the very first time that we start. So unlike in that lawn of cells where you are not sure that you've derived your clone from a single cell, you can actually confirm that upfront using our technology. As you can see here in 2 different examples, cell raft A and cell raft B, There's a single cell at day 1. Those cells begin to grow by day 15. And you can see at the top an example of a monoclonal colony where the entire colony itself is Tra160 positive. In contrast, on cell raft B, you can see that the reprogramming has not been quite as efficient, and in this particular raft at day 15, you can't really differentiate what is on this raft from a bright field perspective. However, when we live stain for pluripotency using a Tra160 marker shown here in the green, you can see that we have a pluripotent colony that could also be isolated. So the nice thing here is that you have the option. You can isolate only pluripotent monoclonal colonies, or you could recover polyclonal colonies and then clone them later on. But it gives us a degree of record keeping and awareness of where those cells derive from that is not possible using other technologies. It was important to us when we were validating the workflow to confirm that our cells were in fact viable despite this new method of retrieving these rafts from the population as opposed to manual scraping. What you can see here in these bright field and fluorescent images is that the reprogrammed colonies were transferred attached to that growth surface. So you see that square here in the image. That is the transfer mechanism that moves the colony intact over to the collection plate. The colonies are transferred without mechanical or enzymatic perturbation. No triple, no EDTA, no 27 gauge needle, no scientist hunched over a microscope inside of a BSc. As you can see, we have that beautiful canonical morphology where the cells are very tightly packed with a high nuclear to cytoplasmic ratio, no differentiated edges along the colony, and in fact, the cells are still pluripotent post-isolation. We then wanted to prove that those clones that we expanded were in fact monoclonal and could be differentiated into the 3 lineages to demonstrate a high degree of pluripotency. So you can see here the single cell here in the center on the raft, and that cell begins to divide and then forms a beautiful pluripotent colony after about a week in culture. We used a canonical differentiation kit from Stem Cell Technologies to differentiate our reprogrammed cells into endoderm, mesoderm, and ectoderm. And you can see that those reprogrammed clones are in fact monoclonal. They maintain pluripotency and they have a high degree of differentiation potential as measured by antibody staining for CXCR4 for endoderm, CD56 for mesoderm, and CD133 for ectoderm. We also wanted to show that the reprogrammed clones were viable and genetically stable. This is a hallmark of the reprogramming troubles that most people face, in that even if you can derive a stable clone, a lot of times you have very common genetic mutations as a consequence of that reprogramming. And so what you can see here is the reprogrammed clone I just showed you that has been analyzed with the human pluripotent stem cell genetic analysis kit from Stem Cell Technologies. And as you can see here, for 2 of the reprogrammed lines in green and blue, compared to the control sample provided with the kit, we have chromosomally normal iPSCs that were in fact reprogrammed without genetic mutation. So to summarize how we can accelerate and automate iPSC reprogramming using our technology, we can achieve 2 times the amount of reprogrammed foci starting from the same cells. So in the workflow I showed you, in both cases, 100,000 cells were transfected, and yet using our consumable, we had twice as many potential reprogrammed clones. When we get to the monoclonization step, we are able to interrogate almost 200 times the number of cells compared to a traditional limiting dilution workflow. In the comparison that I just showed you, we limiting diluted 300 cells in 96 whole plates compared to screening 50,000 potentially reprogrammed cells on a single cell raft array. We were able to identify 336 clones on our system compared to the 1 surviving colony in the traditional workflow in our hands. And what that translated to was the ability to characterize iPSCs up to a month sooner, significantly reducing time, consumables, material, and labor for iPSC development. So once you have reprogrammed your iPSCs, the next step is to not just differentiate them into those 3 lineages that I showed you, but it's to ultimately terminally differentiate them into one of the lineages and a terminal endpoint for whatever tissue type you're interested in studying. That could be neuronal, lung, cardiomyocyte, etc. But the problem with the vast majority of these workflows is that, again, they suffer from the same exact features as all of the other iPSC workflows. Namely, they're manual, you get a high degree of heterogeneity, very low survival rate, and a difficulty to scale. So we wanted to see if we could take those cells that we had reprogrammed and optimize some of these canonical differentiation workflows in order to improve the success rate. So for today's webinar, I'm going to just finish out by showing you 2 examples, one in 2D culture using hematopoietic progenitor differentiation, and the other in 2D and 3D differentiation into neuroprogenitors. So if you have done this workflow before, this may be familiar to you. If not, I'll show you sort of the broad way that these iPSCs are differentiated into the hematopoietic lineage. The key feature of this workflow is that in order to differentiate into HPSCs, you have to generate these very small aggregates of iPSCs that are of a specific size and dimension between 100 and 200 microns. You then go through about 2 weeks of media changes in order to complete the differentiation process as you are changing the growth factors in that media kit to enable the differentiation. Now, the problem here with the workflow comes in this very first step with the aggregation stage. Like I said, the aggregates must be between 100 and 200 microns. You can only mechanically transfer between 16 and 40 aggregates per well of a 12-well plate. And less than 50% of what you transfer typically survives to past the first 24 hours in culture. And unfortunately, any deviations in any of these conditions, namely bigger or smaller aggregates or too many aggregates per well, typically results in the ultimate failure of this differentiation process. And we particularly chose this workflow because a lot of companies and a lot of labs are trying to use HPSCs as the genesis for downstream therapeutics or cell line therapies. So what you can see here on our array compared to a 12-well plate is that each of those monoclonal iPSC clones or colonies is now individually segregated within a microwell. And I call this the Goldilocks experiment because, as you can see here, there are very different sizes of the colonies growing, even within one small field of view. I do want to kind of orient you. You're just looking at a small field of view from the entire array. This particular consumable has over 20,000 positions, meaning that or over 10,000 positions, meaning that there are literally hundreds of these to look at. As you can see here in the red boxes, these are some examples of a colony that we'd consider to be too small. If you look up here in this top red box that just highlighted, that's an example of a colony that would be too big. And as you can see here in the green, this is a colony of the desired size between that 100 and 200 micron optimal dimension. Now, like I said, there's 10,000 positions in this array. No scientist would want to look through 10,000 different colonies and try to identify which ones were the right size by eye, and that is where our software-guided colony size selection comes in. Using our Brightfield-driven software, we're able to use the images that we generate on our platform to identify positions that have single cells on the day of cell seeding. We can also define the size of the iPSC colonies that we're interested in at the end of the growth period, namely between 100 and 200 microns. And as you can see here in this Venn diagram, we can overlay those 2 populations to identify monoclonal colonies of the correct size for recovery. As you can see here in this image, this is an example of what one colony looks like. And in this particular array, there were almost 700 colonies of the correct size. We can then transfer those colonies to a downstream collection plate and continue the differentiation into pluripotent stem cells. So as I'm showing you here, this is the result of that experiment. On the top, you're looking at a clone that was recovered that was less than 100 microns. And as you can see here, by day 7 after transfer, we fail the differentiation process, as you might expect. In contrast, if we isolate colonies that are the correct size, so greater than 100 microns, you can see that as the colony begins to grow, the cells continue to differentiate off the plane of the plate into that suspension culture that is hallmark of the iPSC differentiation. And in fact, by day 17, we have hundreds of thousands of differentiated pluripotent stem cells. And what I want to highlight about this particular image is that both of these colonies started from a single cell. So if you had attempted to do this simply based off of single cell transfer or kind of colony independent of size, this really highlights how these workflows can fail. So simply by tracking that colony size, we're able to improve the efficiency and the efficacy of that differentiation process. You can see what that looks like here in terms of quantitation. Using the automated method, after 48 hours post-transfer, we had 100% survival of the colonies that were transferred compared to only about 10% in the manual method. And in only 2 weeks, we were able to go through an over 100,000 times expansion of clonally derived CD34 positive HPCs in just a little bit over 2 weeks. So to summarize the hematopoietic progenitor workflow, we are able to do automated colony size selection. We can use our software to detect specific sizes and only isolate colonies of interest. We don't need to do any more manual counting or seeding necessary, which leads to a higher survival rate and ultimately the ability to derive not just hematopoietic progenitors, but also clonal hematopoietic progenitors for downstream use, because we can monitor the differentiation of these cells all the way from the derivation of a single iPSC. So the last example I'm going to talk to you about comes to us from a collaboration with our colleagues at North Carolina State University. This was in collaboration with a graduate student who was trying to generate a custom edited iPSC line in a rare disease model. This grad student had been struggling with the workflow, as you can imagine, over the course of her career, and had been unable to derive an edited iPSC line for her downstream differentiation. The challenge of her workflow is that not only were the cells not surviving traditional limiting dilution type methods like manual dilution and FACS, the other main issue is that her edit of choice only turned on when the cells were fully differentiated, which means she had no ability to screen them up front prior to undergoing the various downstream characterizations. So she came to us in the form of a clone challenge in which we help scientists in the community with their workflows using our technology. And instead of going through this laborious workflow that you see on the top, we used our technology to edit her cells, monoclonize them, and then ultimately differentiate them. So what you can see here are the results of that effort. We first took her iPSCs and seeded them as polyclonal in order to maximize the chances of us recovering positive edited iPSCs. That was a necessary step largely because it was a huge construct and the cells had a very low survivability and a very low penetrance of the phenotype. Once we had identified populations or pools of the iPSCs that were positive for her transgene, we then monoclonized them using our technology and to create the monoclonal colonies. Once we created the monoclonal colonies for a downstream analysis, we returned them to the grad student and she performed genomic DNA PCR to confirm which of the ones were edited. You can see here what that looks like in the bottom. There's the single edited iPSC at day 0. We can follow its growth out to the generation of a clonal colony. And when we did the sequencing of these cells, we were able to achieve 17 edited monoclonal cell lines in just over a month compared to almost a year that she had spent trying to generate this line. The real test, though, of the line was to confirm that we were able to see the presence of her GFP transgene when the cells were differentiated. So we did this in 2 ways. We did this in 3 dimensions, which you see here, as well as in 2 dimensions. So the 3-dimensional differentiation was also achieved using our array and our instrumentation. You can see here at day 0, the single cell. We perform the neuronal differentiation directly on the array in an extracellular matrix, and you can see the canonical formation of an embryoid body as we go through the cerebral differentiation process. On day 10 of the differentiation, we transfer that organoid over to a collection plate, which you can see here at day 11, at which point the organoids can continue their maturation. And we were really quite blown away and enthused to see that as the cells begin to differentiate from those iPSCs, we do in fact see the transgene turn on that we expected. We were also able to confirm this in the two-dimensional differentiation workflow, in which the parental and the reporter cells are shown here, where the parental cells are above expressing TUJ1, and the reporter cells, as after differentiation, are also expressing TUJ1, but also the expression of that region of interest with the reporter that we knocked in. When we took those organoids that I showed you previously, the grad students sectioned and stained them. And again, very similarly, you can see the presence of neuronal cells in these sectioned organoids that are in fact positive for the gene that we knocked in, as well as terminally differentiated into neurons. So hopefully in this brief data review, I have piqued your interest in the advantages of automated versus manual stem cell culture. We can do reprogramming on the platform with a high degree of efficiency. We can screen over 150 times the normal of stem cells compared to other platforms, and we can characterize those reprogrammed cells up to a month sooner with less time, material, and labor. We can also expand anything that we have reprogrammed or edited, which eliminates the use of reprogramming with pruning or dissection, with high viability, and we can do that both in 2 dimensions and 3 dimensions of iPSCs and iPSC-derived organoids. We can also edit those stem cells. As you can see in the last example, we can perform rapid screening and edit even rare cells or rare edits, and we are able to do visual monoclonal conformation. And lastly, I've shown you 2 examples of how automated differentiation can increase the homogeneity of your population with fewer cell manipulations and transfers, and ultimately improve your phenotype in 2 dimensions and 3 dimensions. So by now, I'm sure you are wondering a little bit more about this technology and how it actually works. So I'm going to give you a few slides of introduction to our actual technology and highlight how it can benefit you in all of your stem cell workflows. The key to the technology is our consumable, the cell raft array, shown here. It is that multi-well, micro-well grid that I've shown you several times throughout the talk. Within that multi-well array, we can seed our iPSCs, whether they're edited or reprogrammed. The cells settle by gravity into the individual microwells and attach to that square that you've seen, which we call a cell raft. The cells remain attached to the cell raft, and they can continue to grow there until a colony has formed, at which point the raft can be retrieved from the array, picked up by a magnetic transfer wand, and the raft containing your precious cell cargo can be deposited into a 96-well collection plate containing your media of choice, where the cells will continue to grow over time. This is all happening fully automated on our instrument, hands-free, while you are at your desk enjoying that coffee or tea Lisa talked about, and we can transfer up to 96 wells into a single plate, up to 96 rafts. What that translates into is a very streamlined cell line development workflow. All you have to do to get started with our technology is prep the array. You can coat the array with a variety of different coatings and then seed your cells directly into the culture media here. We then use our instrument, the cell raft array, to perform image acquisition. As you saw in some of the previous slides, we are able to take time course images on the platform of every single position in the array, every single day, every single time, while maintaining spatial position accuracy. Imaging is very rapid. It only takes about 10 minutes, and we can do that in 4 colors. You can see what this looks like here for that track and traceability I've shown you several times before. Because the cells are all growing in shared culture media, we have a superior survival of single cells. For the most part, somewhere between 85 and 95% of the cells that attach as singles will grow into viable colonies. We can perform rapid imaging in bright field and red, green, and blue fluorescence, and we can provide a track and trace record of cell growth, which is an image-based record that can be useful for data collection as well as for record keeping for filings. After we acquire those images, we can perform image analysis. This is fully offline using our proprietary software, and it frees up the instrument for use by other users and lets you only be in the lab when you need to work with the instrument itself. As you can see here in a small subsection of the array, you can very easily use our software to identify single cells, as you can see in the green boxes. We can also use the software to measure growth rates and identify phenotypes of interest, whether it's GFP or morphology or marker expression. We can quickly identify the rafts that contain, for example, a single cell at day 1 and a colony at day 3, and which of those are in fact monoclonal GFP positive clones. And then once we have that triaged list, we're able to very quickly map them for isolation using our instrument. That instrument is the cell raft air. The raft isolation, as I showed you in the cartoon, is fully automated. It's compatible with 96-well plates. And the key feature here is that the technology is fully fluidic and enzymatic free. So there's no sheath fluid, there's no tubing, there's no scraping, pipetting, dissociating. That is why our success rate is so much higher compared to other limiting dilution or droplet-based technologies, because we're not perturbing your cells. We're also not moving single cells. When that raft is transferred, we're transferring a colony and not a single cell, so that our iPSCs are never alone in a well any longer, and they're growing as fully validated colonies. The air system is very easy to use, simple operation and maintenance, and it can pretty much be learned very quickly, and you can get off the ground with all of the protocols that already exist in your lab. It's also very adaptable. You can isolate one raft or hundreds. The choice is yours. So within a single array, you may have 600 or 700 colonies. You could transfer all of them, or you could just transfer a dozen. So the choice is yours. And after that, once we've transferred the rafts to the 96-well plate for colony maintenance, typically we see the cells begin to grow within about 5 to 7 days. You can see what that looks like here as the video plays. So what that means is that rather than a plate of cells, single cells in a well, you have a full plate of validated clones. You're not feeding empty wells or dying cells. We have rapid outgrowth, and ultimately what you're left with is a 96-well plate that has your colonies of choice without needing to consolidate dozens of 96-well plates into a single plate of viable cells. This is a very validated technology. We have a lot of customers in the iPSC space, as well as in other cell line development or organoid workflows who have been succeeding with the air system. So we're very confident in the efficacy of these technologies, as well as our customers. If you are interested in an instrument, we would love to talk to you about that, but we also have a lab services arm of the company that allows you to get access to our technology without investing in capital equipment. We can perform custom cell line development, we can help you accelerate your data generation, maximize your team productivity, and ultimately the protocols that we build are yours, and we can help you get a seamless transition to owning the instrument when you're ready to invest in capital equipment after we've done projects with you. If you are interested in learning more, I invite you to visit our website at cellmicrosystems.com. There are lots of application notes and posters and videos that are available to watch to go into more detail in any of these data pieces that I showed you. And you can also go on the website to contact us for requesting a demo or more information. So with that, I'm going to close and say thank you, and I would be more than happy to take any questions.
Thank you, Dr. Hartman, for that fantastic presentation. I loved all the data and workflows and real-world applications. Before we jump into Q&A, we'd like to take a quick poll. If you can answer the question on the screen, we'd really appreciate it. And I realize we're a couple minutes over, so if you do need to go, you can respond to Jessica's email invitation with any questions you may have directly to her, as well as if you have a few minutes to stay on, you can go ahead and submit your questions using the Q&A feature. So let's go ahead and dive into questions. We'll take a few of them for those who can stay on. So to start with, what iPSC coatings can I use on the array?
Yeah, that's a great question. We basically support every iPSC coating that exists commercially. The proprietary nature of the array is very amenable to any coating that you might be interested in. We have validated well over a dozen at this point, and those protocols are available to you. And we provide all of the optimized concentrations and protocols for coating. So it ranges from Matrigel to Geltrex to fibronectin to iMatrix. That's the nice thing about the consumable. It doesn't really require you to change the materials that you're using from a media or a ROCK inhibitor or a coating standpoint.
Okay. Have you optimized other iPSC differentiation protocols on the air system?
Yeah, definitely. For time's sake, I just showed neuronal and hematopoietic, but we have done some lung progenitor differentiation. We have done multiple different types of neuronal differentiation. We have done neural progenitors as well in 2D and 3D. And we have done cardiac as well. So you can differentiate iPSCs into two-dimensional and three-dimensional cardiac tissue. And I'm pretty confident that we could optimize most any workflow with some experimentation, as long as the materials are available to you.
Okay, and we'll just do one more last question. What reprogramming methods have you tried?
Yeah, another good question. What I showed you today was the episomal reprogramming, but we have also tried the Sendai virus reprogramming, and that works just as well. And you can start from either CD34 positive cells or differentiated adherent fibroblasts. Both will work. So again, those protocols are available, and I'm happy to discuss more if there's more specific questions.
All right. As we wrap up today's webinar, I want to extend a huge thank you to Dr. Hartman for sharing her expertise, and to all of you for spending part of your day with us. Your curiosity and questions make these sessions so valuable. Before you go, take a moment to complete the brief 2-question survey that will appear after we end the session. Your feedback helps us as we plan each webinar. And remember, a recording of this session will be sent within a couple of days, and you can share that with others. Have a great rest of your day.
Thank you.